Thanks for having me, Matt. My name is Alex Bisignano. I'm the co-founder and CEO at Recombine, clinical genetic testing company based here in New York City. And I'm going to be talking today a bit about data science and genomics and some of the data challenges that we face scaling up a genomics company. So I think a good place to start, though, is probably genetic testing and how genetic data was historically generated and analyzed. So just to ask a few questions here. Who here has a genetics background in advanced degree in genetics or any sort of, I apologize in advance, I'm going to cover some basic things right now. Who here has taken at least like a basic biology class in high school? Awesome. So we all know what genetic material is. DNA and RNA are the two primary ways of storing information for biological organisms. And it's actually the first digital storage system, which is pretty interesting to think about it from that perspective. What is digital by definition? It means you could actually copy the original without affecting the original. The way DNA is replicated, it actually does not in any way harm or change the original copy of the DNA. So that by definition makes it a digital data storage. And this is a cool DNA picture that is digital. And just sort of to cover genetics 101 in one slide, human genome, we have 23 pairs of chromosomes. One of those pairs is the sex chromosome. So you either have two X's, which makes you female, or an XY combination, which makes you male. The chromosomes are made of nucleic acids. For DNA, there's 4 nucleic acids: adenine, cytosine, guanine, and thymine, A, T, C, and G. These chromosomes code genes. It's actually like compiled code that has instructions that create proteins that tell your body to do things. So there's about 3 billion bases in the combined human haploid genome, which means that there's a total of around 6 billion bases to make up a human being. So that's everything you need to know. You are now all genetics experts. So I want to start with what we already understand. Moving into 2003, where we spent 10 years and $3 billion finishing the Human Genome Project, what do we actually already know about genetics? Aneuploidy is one of those things, which is chromosomal abnormalities. During meiosis, which is the production of sperm and eggs, you can have something called non-disjunction, where you actually get an extra copy of a chromosome or lose a copy of a chromosome. And what happens here, it could cause things like trisomy or monosomy, and these result in syndromes like Down syndrome. More often, actually, extra copies of chromosomes will result in failure to implant or in early pregnancy loss for embryos. And the incidence of chromosomal diseases is about 1 in 500 live births across all age groups, with the risk increasing as the parents get older. The classical technique for testing for this is something called fluorescent in situ hybridization, or FISH, where you literally attach these fluorescent probes to all the chromosomes and you count. It's fairly qualitative, and you can see them under a microscope. Something else we understand is single gene disorders. These are inherited genetic disorders, whereas aneuploidy is generally random events that occur during cell division. Single gene disorders are inherited diseases that can be carried on through generations, and that's actually what we test for at Recombine. Cystic fibrosis is the most common disease found in European individuals. And what happens is carriers of these genetic diseases, the parents in this case, have no symptoms. They're perfectly healthy individuals. If 2 parents both carry these diseases, 1 in 4 of their children will get 2 copies of the broken gene and in turn inherit these diseases. And the classical way of testing for this is polymerase chain reaction and restriction fragment polymorphisms, your classical gel runs. You can see sort of your CSI pictures here. Again, labor-intensive, expensive, and we're looking at a single mutation at a time for many of these. So the amount of data we're generating is 1 for many of these. And what's interesting about both of those types of genetic diseases is they're what we call high penetrant. If you have the mutation, if you have the extra chromosome, you get the disease. There's really no probabilistic way of looking at that. It's about 100%. So what happened in 2003? We finished the human genome, and then for the next decade, the human genome, the cost to sequence it went from 3 billion down to 1,000. We're now generating exponentially more data. So if you took a minute to analyze every piece of data using that chromosome technique, it's maybe 15 minutes to look at aneuploidy. As we go into the whole genome, it would literally take 5.85 millennia to analyze the whole genome if we were to do it by the human eye. That's obviously not how we approach this problem. So we use computers. They're really cool tools. And we still have some data challenges that remain in genomics. Data storage. I mean, we have some great database systems, but standardized data storage system still remains a problem for many laboratories. Most laboratory diagnostic companies are not set up as software companies. Example, Quest and LabCorp. two largest companies in the country that do laboratory testing, $12 billion market cap. They are not agile software teams. Data integrity. There's many databases that annotate the human genome and say things about it. There's lots of differences, lots of inconsistencies. Mark from Solve Bio will be speaking and talking about how they're attacking that problem. It's a real problem. It's something that we deal with every day. And then data sharing in general. Data sharing, we don't have GitHub yet in genomics. There's no good way for collaboration, so we don't have this magical open source movement. Universities are kind of afraid of forfeiting IP, so we've got all these weird things that are preventing us from really revolutionizing the world. But let's move beyond single gene for a second. If we were to imagine where genomics starts getting more complex, it's these complex genetic diseases. How do we model, predict, and clinically act upon things that move beyond the single gene or the aneuploid that I described earlier? So what is a complex genetics disease? It's something that can affect all of us, diabetes, autism. It's things that don't have a single genetic disease, that might have an environmental component, that might have many different factors that lead us to either get or not get these disorders. They can be influenced by a number of things, and it's not 100% certain we'll get them. An example of something we look at Recombine, which has to do with couples undergoing infertility treatments, is the ovarian response to follicle recruitment. We deal with patients that are receiving hormones to help produce multiple eggs in the fertility treatment, and oftentimes we'll want to know how are they going to respond to these drugs. And right now, it's kind of like throwing spaghetti against the wall. They give them a protocol, they see what happens, and then they try again later. It's expensive to do that. And we're working on developing algorithms to predict this ahead of time. But just as an example, if we look at the pathway which is illustrated on this slide, there's clearly a number of genes that act together to ultimately cause an egg to be produced in the ovaries. If we just look at 6 of them and the number of variants that we know of, there's many thousands of combinations that may exist. So our knowledge in genetics up until today has said, look at a single gene, make a diagnosis. We can't look at 10,000 combinations for every patient. That's just not possible. So the paradigm's obviously changing to a more computationally driven analytic focus. One of the other problems we have with complex genetic traits is clinically, how do we deal with this? Medical decisions are, in genetics at least in reproductive genetics, used to binary outcomes. You have aneuploidy, you get disease. You have single gene broken mutation, you get cystic fibrosis. A good example of a change to that paradigm is actually in BRCA screening, and this is for inherited breast cancer. And it's a form of breast cancer that the average patient has about a 12.5% lifetime risk to getting. But carriers of a mutation in the BRCA1 gene have a 60% lifetime risk. One of the problems that we faced as a clinical, in the clinical world is how do you communicate this to patients and how do you act upon it? It still doesn't mean you're going to get it. You can have a mutation in this gene and never get breast cancer your whole life. And it's a change in the paradigm of how we treat patients, and it's something that physicians are just coming around to and just starting to develop good protocols for really optimizing patient outcome. And genetic counselors are actually the medical specialists who really deal with this communication to patients. And right now there's about 1 genetic counselor for every 250,000 individuals in the US. It's a rising need in our medical system with all this data and all these new tests that are being developed. Who's going to actually help patients make decisions based on it? It has to be them, and we need more of them. But ultimately, if we just look at where and how we've discovered things in genetics, we've really started on the outliers, the single gene, the aneuploidy, the really high signal outliers of the population, and those have helped us identify genes that affect many different processes in the bodies, and we're starting to work our way into this distribution of where people might lie for a particular disorder. And ultimately, one day, hopefully, we'll be able to say things about everyone. The idea is that you can go to any medical specialist and you'll get a genetic test that will help them optimize the way you're treated. So that's complex genetic diseases and the outlook we have. I'm going to talk a bit about a specific data challenge that we've seen at Recombine very quickly, just to give an example of something that we've had. So automation of data analysis is very important when you're dealing with that much data. But automating data analysis can be dangerous, especially if you're not planning for your unknown unknowns. And an unexpected challenge that we saw is due to microarray testing. So we use DNA microarrays to test our patients, as well as genetic next-generation sequencing. The way a microarray works is you have essentially this grid, which is on this slide, that has a bunch of wells. And in these wells, you have silicon beads. And these silicon beads have synthetic probes attached to them that are specific to about 50 bases in the human genome. And what we do is we take the patient's DNA, we treat it, and then we expose it to the microarray. The DNA is kind of like a magnet to the probe, and it'll literally hybridize, as it's called, where the genomic DNA, the blue one in this image, will attach itself to the probe, and then we run what's called a single base extension assay. So a single base, a nucleotide, is added to the end of the probe, and depending on what genomic DNA is present in the patient's DNA at that spot, it turns a different color. It actually is fluorescent because it's a synthetic nucleic acid that fluoresces. And it will fluoresce one of many colors. And what we do is we measure the wavelength, we measure the intensity, and we get these nice little mutation clusters, we call them. So this is an example of what we might see if we're looking at a mutation that can be either an adenine or a cytosine. Every dot here represents a patient, and the wavelength is on the horizontal line. The vertical line is actually intensity, not number of individuals. It was a mistake on my part. And what we're seeing here is that when you have two A's, it's fluorescing just blue. When you have two C's, it's just red. But when you have an A and a C for an individual, those light patterns will combine, and we'll actually be able to measure an intermediary wavelength as we combine both of them, which is green, because blue and red make green naturally. And that's how we identify people who have both A and C, because we have two copies of every mutation. So in this method, using microarrays, we can differentiate between two nucleic acids. It could be either A or C, A or G, T or C. We have to know ahead of time what we're differentiating. But when you only have two light channels and there's 4 possible nucleic acids, isn't that a problem? Can't we only determine like 2 of them? So one of the paradigms was we should only test for highly conserved single nucleotide polymorphisms that only differentiate between 2 bases. So that means regions of the genome where we pretty much say that every human being only has either A or T. They won't have C or G. And that's what we were told to test for. But this is an assumption. We can't actually know that every human being on the planet only has A or C, not T and G. And what that led to is a kind of a problem that we saw in a particular disease when we were testing for it. So there's a disease called CANAVAN disease. It's caused by mutations in the ASPA gene. And there's one mutation in particular, 693C to A, which causes a termination codon. It breaks the gene. In the literature, based on what we read, 99% of the population carries a C, which means that they are healthy and they do not have this disease, and less than 1% of the population carries an A. What we found out when we started testing people is that we were seeing much higher rates than 1% carrying this A, because we were really only looking for a C or an A, because that's all we thought would exist at that location. It turns out that the reality is that AG was carried by a pretty significant portion of the population, and I will say it was in specific populations that hadn't been previously very well studied. Most of genetics actually is based on studying European and Jewish individuals, just because that's been the body of literature that's been developed over the past few decades. So we're actually learning a lot about many other populations in the past several years. So this is actually what we were seeing when we looked at our clusters again. You had your AA group, the normal folks who didn't carry the mutation. But then the middle cluster could have been AG or AC, and then the right cluster could have been GG or CC. So this complicated things, and we had to develop a strategy once we saw this to test for every possible combination. And this is important. We realized you can't make assumptions about the human genome because there's just so much we don't know yet, and it kind of illustrates that this is such a young field that we can't possibly even know what not to expect just yet. So ultimately, we took away that assumptions about the genome are probably not correct. As we're generating more data, we're learning that there's more and more variation. We actually haven't truly sequenced the whole human genome just because there's so much variation that we can't capture yet. And even great genomic technologies need sanity checking. So just an interesting real-world example of problems with data as we've been analyzing them in the human genome. So I'd like to thank you for your time. Thanks to the Recombine team. I know we've got a bunch of you guys here in the audience. Thanks for coming to support us. And yeah, thanks, Matt.
So thanks very much for this, and thanks for not turning this into a product pitch, which is something I typically ask people to do, but they don't always do that. But so since you've been so kind from that perspective, could you actually tell us a little bit more about what Recombine actually does, just in a minute, the first product in particular?
Absolutely. So I mentioned single gene disorders like cystic fibrosis. Those are actually regularly tested for in the US by either fertility physician, reproductive endocrinologists, or OBGYNs. LabCorp and Quest, who are the two biggest labs, dominate that testing market. And each disease that you might test for, cystic fibrosis, spinal muscular atrophy, Fragile X, Tay-Sachs, these are well-known diseases, can cost thousands of dollars using older technologies. So the first thing we did actually is apply these microarray and sequencing technologies, and we developed a test that can do 250 of the most common diseases of this nature for under $500 to patients that don't have private insurance. And this is just making it more accessible. It's generating more data. It's more accurate. It's faster. It's really an application of these technologies to an area that we're already regularly doing in medicine. And beyond that, what we can do now is we can leverage the amount of data we're generating at the point of care and actually run research and development and become an asset for a physician. So,
just to take it at a high level for people that just haven't spent at all, time thinking about all of this. So, 23andMe, which is the name that everybody knows in that field, is a consumer-facing product. This is a good doctor
so
prescribed.
This is a laboratory-developed test. The laboratory testing business is governed by a clinical amendment called CLIA. So we have approvals to actually run this test, state approval from New York. We sell through referring providers only. There's medical indication when our test's ordered. What's nice about this is we're actually part of the patient's healthcare process, part of their standard of care. And actually helps us because we're covered by pretty much all major insurance companies. So from a payer perspective, when someone has private insurance, generally our patients don't pay more than $100. It's really making this accessible, and it's really helping to prevent some of these serious illnesses.
And last question from me. So this company is very symbolic of this new generation of companies which are as much software and data companies as they are life sciences companies. How does that translate in your team? Who do you have that does all those things?
It's actually, I love my team. It's an amazing combination of people. We have engineers like any tech company in New York City would have. We have an operations team that is one of the most efficient group of people I've ever seen, from billing, client services, logistics. We had to learn how to track our own inventory. We stock IVF clinics and fertility OBGYN offices with blood kits. We have a laboratory team that they receive samples on a daily basis, extract DNA, analyze it. And we have a bioinformatics team, and what they do is they're sort of that intersection between generating the biology data and interpreting it for the engineering group, and we're building a data science team too. I mean, analyzing this data and actually learning new things from it, being able to look into that complex genetic world and start developing it. It really requires coordination in all these diverse groups of people, and I think vertically integrating that way has allowed us to really control the data and make sure that we're generating clean, good, usable data for our research as well.
Great. One or two questions from the audience, if any. All right. Closest.
It's a great summary of biology. How do you use your data? How do you, I think from all the patients that you collect, is it publicly available? Obviously not, but so is there any data analytics that is shared or published or anything like that?
It's a really good question, and this is an area I think that warrants a lot of discussion in general. I'll sort of be brief. Utilizing data from clinical samples is something that some groups are very progressive. They make it publicly available. We have patients opt in to research. And there's a general research which allows us to use their data aggregately in a de-identified manner. When we start using it for anything beyond that, we actually obtain what's called Institutional Review Board approval for any specific studies in human subjects. And generally that's required if you want to take anything through an FDA process that you analyze. And ultimately, we're not necessarily being conservative, but we're following respect for humans and general guidelines for how you use human data. We see a 65% or more opt-in from most of the centers. If patients are fairly empathetic, we don't make it publicly available just yet. I don't know if that's a route we'll necessarily go down. It's certainly something we use for internal research, and we've published in major conferences, generally looking at what are the carrier rates that we're seeing versus historical literature. So definitely we've used our data to open up conversations around screening as well.
Good. One more. So I think Andrew is still there. Do you have a mic, David? No? Okay, can you? So I guess, yes.
No, biotech background, so bear with me. But are your techniques similar to what companies have been doing? Is you're just using better machines, or is it the technique itself is very different? And if it is, can you talk about the accuracy rates of what you are producing versus what the other companies are producing? And of course, the related question that if accuracy is better, why would anyone test through those previous methods, which are costly and time-consuming?
That's a really loaded question. So I'll tackle it by piece. So accuracy, we've actually tested our own and developed our own ways of testing for these mutations. The development of our actual core technology is based on a platform technology company called Illumina. So we've developed on top of their platform a custom version of their platform that we then use our own custom algorithms to analyze. So it's a combination of really good molecular design as well as really good software. So that's sort of the secret sauce. The way we validated ours has shown greater than 99% sensitivity and specificity, which are the standard of care. Why are people using other things still if this exists? We all know healthcare. We all know insurance companies are a big part of that. LabCorp and Quest have sort of an oligopoly on insurance contracts, and people tend to stay in network. And we now have a national sales force that is literally going out and teaching physicians about us and how even if you use this out-of-network laboratory, which we are, it's still less costly to the patient than your in-network laboratory. So it's a lot of physician education. I think that's starting to get that change to happen. But certainly the payer situation keeps people on older technologies.
Great. Well, thanks very much, Alex, for coming and talk to us.